All right. Okay. So this is what I talked about yesterday. Ooh, turn that off. So, like I said, glycolysis, we can use all kinds of other fuels besides glucose. So, polysaccharides can be broken down into monomers, and things like glycogen or starch, the monomers are glucose, and they can just enter right at the beginning. Other 6-carbon sugars can be modified, taking off functional groups, moving things around to make glucose. So galactose, fructose can be modified to start at the very beginning of respiration. Okay, so proteins have to be broken down into their amino acid parts, and then we don't want that amine group, so we have to get rid of that. We'll take the carbon chain, modify it however it needs to be modified, and depending on what you have left, you can enter it different parts of glycolysis or in the Krebs cycle. That amine group will go off and will form urea. So, so fat will break down the glycerol off of it, and that can be utilized in the fatty acid. Will, and like I said yesterday, the glycerol will be converted to D3P and enter that fifth step of glycolysis. And the fatty acids, look at all those acetyl-CoAs. Whoa. Okay. So each one of these parts of a fatty acid we turn into acetyl-CoA and will enter the Krebs cycle. So if you actually look how many acetyl-CoAs you can make from just this one fat, it's a lot, which is why fats are way more what we call energy dense than carbohydrates are. You can store a lot more energy in a small amount of space. Look at all that. That's a lot. So that's what I just talked about. All right, now this whole process needs to be organized and it needs to kind of all run at the same rate. So there are ways to kind of check it to allow it to slow down if a part needs to catch up with another part. Oh, excuse me. Or whatnot. Hold on. Hold on. Everything can go backwards, by the way. Every reaction is catalyzed by an enzyme. The enzymes can go both ways, just FYI. It can go backwards if necessary. And these images are basically from biochemistry textbooks, but this one line here, like here's the Krebs cycle. So there's a lot of different ways that carbohydrates can be metabolized in the body. Same thing for lipids. So there's the Krebs cycle right there, but look at all the other reactions that can take place. And then this is amino acid. And there's the Krebs cycle. Anyway. So, all right. And this is nucleotide metabolism. Again, here's the Krebs cycle. It's complicated. Excuse me. Okay, acetyl-CoA is super important. And here we go. So let's talk about feedback. And the only enzyme, like I said, you need to know is an enzyme in glycolysis. It's actually called phosphofructokinase. Phosphofructokinase, there it is, it's in purple. All right, so remember how we talked about negative feedback before? Yeah. Permanent situation, it'll pop back off. It's important that it does pop back off so that pathway can continue when it's needed to. And remember, you can increase the inhibition of it by just having a lot of the product. But if you start to run out of the product, so it pops back off, it's unlikely that another is going to bind right away. So the pathway will continue. So, like I said, phosphofructokinase is the enzyme, the major enzyme that is regulated in all of cell respiration. It's an enzyme in glycolysis. And it's important that it's here because this enzyme is, if you look at I'm going to skip over to this other PowerPoint. Here's phosphofructokinase, right here, number 3. So it's the enzyme that occurs that catalyzes the phosphorylation of this fructose 6-phosphate to make this little guy over here. As soon as this guy is phosphorylated to make this molecule, fructose 1,6-bisphosphate, is super unstable. It has those 2 phosphate groups that really wants to just break in half. So if you allosterically inhibit this, it prevents it from being phosphorylated again and prevents it from eventually splitting. You're just stopping it there. At this point, it's kind of really unstable and it's going to break. So that's why. Prevent it from moving, the point of no return kind of thing. So let's look at it. The things, if you look at the green line up here, AMP and ADP increase, will stimulate it. And why? Why? Does anybody know why? Why would those stimulate it? Yeah. If you use a lot of your ATP in the cell, then you're going to have a lot of ADP. And even ADP can be used and pop up one of its phosphates to make AMP. So if you have a lot of AMP and ADP, that probably means you don't have a lot of ATP, which means you need to get going faster to make more. Do what?
So then, what do they do to the enzyme that stimulates it?
It can increase the binding, make it go a little bit faster. There's ways that happens as well. All right, ATP, however, will inhibit it because if you have a lot of it, you don't need to go quite so fast. What's interesting is that citrate can also inhibit phosphofructokinase. Now, if you have a lot of citrate, that means you're breaking down the glucose really fast. You're making a lot of pyruvate. You're making a lot of acetyl-CoA. It's binding to oxaloacetate, making a lot of citrate. So if you have too much of it, you're going to be making all these electron carriers. But if it's all backed up, they can't drop their electrons off as quickly as they need to. So citrate, the reason that it's an inhibitor, it kind of works to make everything go at the same pace, basically. Okay. So why is it important? So the cell doesn't waste its energy. Okay. Oh, you don't need to know all that. All right. Let me talk about what you're doing today. So if you look in your packet, and I have mine right here, you have, it says on the front, building the model. So this is basically the chalk drawings with a lot of questions, okay? This kind of tells you or gives you an idea of what you're supposed to be doing. Use chalk on a tabletop, which is what you're doing, so not a marker on a sheet of paper. It says draw the cell membrane and the mitochondrial membrane. And you're using cutout pieces of paper, not Play-Doh, okay, to represent the molecules, ions, and membrane transporters or pumps. Okay, you'll use all the colored paper I have in the back. Got it? Ions. What kind of ions do you have? Hydrogen ions. You have electrons. Okay, you'll be using electrons on these guys too. Molecules and then the membrane, like ATP synthase, all that. Then it says, use the pieces you assembled to model the process of fermentation and aerobic respiration. Excuse me, develop a dynamic model that allows you to manipulate or move glucose and its breakdown products through the various steps of fermentation and aerobic respiration. When you feel you develop this good working model, you have to demonstrate and explain it to me. You'll be in groups, okay? When you group present to me, one person cannot do all the talking. If one person does all the talking, I'll be like, I'm coming back. You have to share responsibilities because I need to know that everybody knows what you're talking about. Got it? I don't care how you split it up, but you all need to make sure you know all of it. They kind of give you some things you need to make sure you include. So it says, be sure your model of fermentation includes and explains the actions and roles of glycolysis, cytoplasm, electrons, protons, glucose, NAD+, NADH, ADP, phosphate, inorganic phosphate, ATP, pyruvate, ethanol, or lactic acid. You pick, you don't have to do both. Pick one, okay? And then substrate level phosphorylation. That just means you have to explain that part to me, like what is that? And then on the back it says, be sure your model of cell respiration includes and explains the actions and roles of glucose, oxygen, CO2, pyruvate, acetyl-CoA, NAD, NADH, FAD. Y'all, Mr. Jesus' video had a few errors in it. And I'm just saying, if you notice them, they're there. He put FADH instead of FAD. Did anybody notice that? Okay. I'm just saying, if you want to use this video to study, just know that there are a couple errors in it. FADH2, ADP, phosphate, ATP, water, ETC, mitochondria, inner, outer membrane, hydrogen ions, electrons, chemiosmosis, ATP synthase, cristae, proton gradients, oxidative phosphorylation, substrate level phosphorylation. They include that twice, didn't they?
Yeah.
All right. So, yeah. You'll need to make sure you clean everything off, like remove everything from your bench, because you're going to need to use an entire half a bench for your stuff. Okay? If you don't get to present to me today, then I'll give you the beginning of class tomorrow to present to me. Okay? It probably is going to take you at least 2 or 3 times before you get it right to me. That's just the way it is. Don't stress about it. As soon, like I won't give you a grade until you get it all right, pretty much. Now by the 3rd time, I expect you got it right. If you don't, you might have some problems. But, and then you're going to answer all of the questions in this packet, and you'll eventually turn this in probably by the end of the day tomorrow. Got it? Okay. There's chalk back there. What else did I want to tell you about?
So we're drawing out fermentation as well as cellular respiration?
Do all of it.
Okay.
Yeah, yeah. And you can do it all kind of on the same area. Like, you know, fermentation occurs in the cytosol. So you can have like glycolysis and the fermentation over here and then the mitochondria there. You don't have to worry about like the nucleus, any other membranes at all involved. We just really care about the mitochondrial membrane. Also, you're going to need to use your textbook because this is where these diagrams are really going to help you. A lot of times, now let me talk about this before y'all get going. Like this is not enough detail. Okay? I don't want you to draw all these figures, but basically what I want you to do is have some sort of you're going to start with glucose as your beginning product or beginning reactant, okay? Glucose has how many carbons? Basically, you're going to need to use the carbons and like phosphates and show me what's happening at each step. You don't have to name these things, but you have to show me. So you'll have some sort of 6 carbon thing. I've had people just do like, don't have a ring structure necessarily, but they draw like C, like 6 of them. And then they have a little phosphate they can pop on or whatever. And then they'll rip it in 2 to have 2, 3 carbons. Does that make sense? Okay, but you need to kind of show me what's going on at each step. After the 5th step, hey, are you finished? Okay, hold on just a second. It splits into 2, right? And then you need to make sure, here are the next 5 steps that you talk about. You need to have a little NAD plus, little electron carrier bus thingy thing that you can drop electrons and hydrogen onto to make an NADH. Some people have a card where they have like NAD plus and they flip it over and there's an NADH. You know, do whatever you want to do, be creative, but these things need to be movable because the NADH actually will leave and go to the ETC. You have to be able to drop electrons off. Electrons, you need to be able to show them moving through the protein carriers. You need to show how hydrogen ions will pump through, like all this stuff. It's going to take you a while. Okay? Yeah.
So like separate fermentation and cell respiration, do you want us to like get into fermentation and then talk to you and then start the cell?
You can show me all at once. Okay. All right. You can pick your own groups. I don't care, but I want groups of like 3. That's pretty much all. If you need the only 2 groups per like big lab table, if you need to go on the side, that's fine. Just kind of move stuff around. There's some research projects going on. Just be mindful of that and don't mess them up, okay? There's shrimp in the back and stuff, so.
Shrimp?
Yeah, brine shrimp. They're tiny. All right, go. You pick your own drinks today. And you guys have your book. I love it when my
Let's go. Let's get my picture for it. Hey, look at this one.
Yeah, where's the chop?
Don't put it on the table.
You guys want to share the chop?
Yeah.
You gotta make them think. Yeah.
I think I might find Oh wait, no, I have. Why are you here?
Y'all get this over there.
Hey now. Ask me for what? Go to the house.
I feel like you're gonna. I have the.
Oh, it's over there. It's 158.
I know, but like, we have to do both. I think.
How many?